Yeah, it should be. Hey guys, good. How are you? Hey. Yeah. Good. How are you? I don't know. Like, honestly, there's. Yes, so that is the one. Yeah. No, no, I mean, yes, but yeah, man. Who's not here? Who is not here? Yeah. I will. Yes.
Okay, we'll get started as soon as two of the committee members show up. If you guys need any questions, help yourselves. We have a few minutes. Okay, Lourdes is here. Okay, I think we should start. I'm sure Dr. Cohen wouldn't mind joining us a little bit later, whenever he arrives. It's a pleasure to introduce Sebastian Vega, who, as you all know, is a very active member of our community here in chemical engineering and biomedical engineering. Sebastian came to us from Carnegie Mellon University as a graduate in chemical engineering several years ago, and has been an integral part of the MOGI Lab, part of the IDER program. He's also been very active in terms of mentoring, especially underrepresented minority students. He's been part of our diversity ambassadorship, which is a network across the US of STEM graduate students that would go and mentor other students elsewhere. And finally, he's gone abroad as part of the IDER program. He spent a summer at the Twente University doing research with Jan de Boer as well. So it's been a very eventful journey. And he's going to take about 35 to 40 minutes of your time to summarize his research to date. Then you can ask him questions. Then we'll have you excuse us. The committee members will stay and deliberate a little bit more. Sebastian, take it over.
Thank you, Dr. Mogi, for the introduction. So I'd like to thank my committee members for coming in. I would like to also thank all of you for coming and spend your time to hear my talk about my dissertation project. So my dissertation project focuses on single cells and profiling of nuclear proteins in order to identify different cell states of stem cells. So stem cells are a viable tool for therapy and research, and they are multifaceted in terms that there's many different types of research being done in therapies. So for example, they are good cell source for disease models and drug screening. So for example, if we take stem cells and we can differentiate into different types, we can unravel molecular mechanisms of different diseases involving cardiovascular or neuronal related. And then also we can use stem cells as a platform for drug screening. So we can test the efficacy of different drugs, and we can also do toxicity or liability tests to assess the response to those drugs. And if those stem cells are gathered from the patient himself or herself, Then we could do kind of like a human preclinical trial too, and then at the end of the day, develop new drugs that are completely personalized. In addition to that, stem cells alone serve as a viable source of different cell therapies. So one of the more common ones are bone marrow transplants. Here's a schematic of allogeneic bone marrow transplant, where a recipient that needs bone marrow, either due to certain blood disorders or cancers like leukemia, gets a matched bone marrow from a healthy donor. And last but not least, another application of stem cells is to engineer tissues and organs for transplantations. So here's a sample workflow of how this could work. We could take stem cells from a patient, we isolate them, expand them, put them in the right microenvironment, which is biomaterial properties, growth factors, culture it in a bioreactor, and then put it back in the patient. So this type of workflow has been successful for things such as making bronchial tubes and bladders. So there are many types of stem cells, but there are three main categories. So the first one's adult stem cells, which are everyone in this room has them, and they are typically derived from the bone marrow, but they can also be gathered from adipose tissue. And these are advantageous because they're autologous, so they come from you. Hence, if they were to be used for some therapies, there would be no negative immune response. However, some of the limitations of adult stem cells is that they are multipotent, meaning that they can only differentiate towards a handful of lineages, so they cannot differentiate towards every cell type in your body. And hence, these are good for quote-unquote simple engineered tissues, but not for more complex organs. So to address that limitation, there is embryonic stem cells, which are gathered from the blastocyst from a recently fertilized egg. And then these are advantageous in the sense that they're pluripotent, meaning they can differentiate into any cell subtype in your body. And however, one of the limitations is there's potential immunological response since these cells don't come from the patient. And there's also ethical concerns having to do with the destruction of an embryo to gather these cells. So to get the best of both worlds, recently it was discovered that if you take an adult somatic cell or a fully differentiated cell, for example skin cells, you expose it to a small cocktail of transcription factors, you can reprogram the cells so that they go from fully differentiated to a cell type that behaves like embryonic stem cells. So by doing this, you get the same properties of embryonic stem cells. These are pluripotent cell population, and they're also autologous because they come from the patient, him or herself. However, these also have some limitations. Mainly, in order to reprogram these cells, you need to introduce some genomic modifications, which is not very attractive for clinical relevance. So, even though stem cells they have huge potential for both therapies and research, there are some limitations. So we said if we take stem cells and we expose them to a certain microenvironment, which consists of biomaterial properties and growth factors, they will, under the right conditions, differentiate towards different lineages. And the two key challenges here that need to be addressed to further the research and therapeutic use of stem cells is first stem cell derived populations of heterogeneous. Meaning if you have a population of stem cells, they're not all going to behave the same way, which is a huge problem. And another one is that there's a lack of early markers to measure downstream stem cell differentiation. So the differentiation process is very dynamic and lengthy, and there's what's going on between initial cell contact with the microenvironment and the eventual phenotypic output is not well understood. So we'd like to address these limitations by employing imaging-based strategies. So one of the studies that motivated a lot of my work come from the study here, where they showed that if you take an adult stem cell and you expose it to growth factors that either allow it to become a fat cell or a bone cell, but you restrict the area, so you put them in a tiny area, the cells will tend to become preferentially become fat cells. However, if you put them in a larger area and allow them to spread, they tend to become bone cells. And the authors of this study also showed that there's a role of actin that has to do with this, or early changes in actin, and also Rho-A-induced actin polymerization. And in a more recent study, actually looked at the same area, so we restricted cells to the same area, and then either put them in, restricted them to a round morphology or a dumbbell-shaped morphology, and they saw that cells in this morphology preferentially became bone, and the ones in this preferentially became fat. And then the authors also showed that actin was a key player in all of this. So this motivated us at the Moge lab to develop this workflow using actin as a predictor of future stem cell responses. So what we did here was we took mesenchymal stem cells and then we put them either in an adipogenic induction condition, which gives fat, or an osteogenic induction condition, which gives bone. And then at very early times, so within the first 96 hours, which is before the cells will actually differentiate, we fix the cells and then we stain them with actin, and then we gather individual cell segmentation. So for each individual cell, we gathered a set of morphometric features that are descriptive of the morphology. I will refer to these as descriptors for the rest of the talk. So once we gather these descriptors that describe the shape, the intensity, and the texture of actin, we reduce the many descriptors to three-dimensional space to see if we can identify morphologic differences between two conditions. So by doing so, as early as 24 hours in the presence of fibronectin, we're able to identify morphological differences between the bone condition, which is blue, and the fat condition, which is red. So here in this plot, each dimension is a combination of the descriptors that describe actin. And each of the dots represents a cell, either in the osteogenic bone condition or the adipogenic fat condition. So here we see some nice separation. However, in the absence of fibronectin, that is not the case. We cannot separate the two for 24 hours. Then we did a genetic study, and then we found that in the absence of fibronectin, at 72 hours, we started to get some nice separation between the two conditions. So even though this is very promising, there are a few limitations for cytoskeleton-based profiling. So the first one is that we need to segment individual cells. And this is problematic if you have many cells. So if we were to want to study the effects of this particular material, or you just have a topography that you're interested in, and then you put the cells in there, and then you do your single cell segmentation, and there's too much cell-to-cell contact, you can't really tease the cells apart. So there's really no way of really telling how many cells we have here. And then if we stain them for the nuclei, we see that there's 10 cells in this picture. Another key limitation is that the challenges in order to further the use of stem cells in therapies and research have not been addressed. Mainly the ability to identify cell subtypes and heterogeneous groups, and the development of early screening tools to predict biomaterial responses in terms of stem cell differentiation. So in order to understand this, we have to take a step back and then, instead of looking at actin, potentially look at other proteins in the intracellular space that are also affected by actin. So here is to reiterate, if you culture cells and a certain set of material properties and soluble hues, the cell will differentiate into different lineages. But if we take a closer look, we know that actin is a key player from previous studies and our own research. But there's also, actin is modified by coadhesions and integrins, and then in turn actin affects the nuclear space. So we refer to this as an outside-in signaling that emanates from the outside of the cell via actin to the nucleus, which is the central hypothesis of my dissertation, which is that there's a physical continuity between the environment and the nucleus mediated by actin. Brings me to three specific research aims. So the main goal is to expand the utility of single-cell imaging-based profiling. And to do so, the first aim is to develop an imaging-based approach to classify single cells using intranuclear reporters, mainly proteins and molecules in the nucleus so that we can look at confluent populations. And then once we do that, the second aim is to identify distinct cell subtypes and heterogeneous groups. And then the third aim will be to utilize this tool that we developed in aim 1 to predict based on a set of morphometric descriptors. So we identify future responses to biomaterial conditions. So for the first aim, we sought to identify, well, modify our profiling approach and also identify nuclear reporters. So here we culture mesenchymal stem cells in glass, and we expose them to either fat, growth, or bone growth factors. At 72 hours, we really see no differences. However, if we culture the cells in the same conditions for 2 weeks, we see differences. In terms here, the green are intracellular triglycerides, which is a marker for fat, and then here the blue is intracellular alkaline phosphatase, which is an enzyme that's highly expressed in bone differentiated cells. So the question or the objective of this aim is can we identify nuclear proteins that are distinct between different microenvironments that can before the 2-week differentiation outcome. So to do that, we looked at a small cohort of candidate nuclear reporters, and first we looked at gene-modifying reporters. So the first one here is histone-free trimethylated the 4-glycine residue, and then this results in an open euchromatic state of genes that are necessary for osteogenic programming. And we also looked at EZH2 homolog, which is a component of the machinery to trimethylate histone-free of the 27-glycine residue, and that results in the eventual decrease in osteogenic gene expression. And then we also looked at RUNX2, which is a transcription factor that is necessary during the early stages of osteogenesis. And the second family of nuclear reporters we looked at were mechanosin stuffed. So first we looked at laminin, which is one of the key components of nuclear lamina, and is very closely linked to actin, which we have shown actin is very responsive to the microenvironment. And then within the nuclear space, we looked at 2 other proteins, mainly nuclear mitotic apparatus, which several studies have suggested that it's one of the key components in the nuclear scaffolding matrix. So it kind of helps hold all of the compartments in the nuclear space together. And then we also looked at SE35 domains, which are interchromatin domains, meaning they reside between chromatin regions. So they may serve as indirect measures of global chromatin changes and global changes in the shape of the nucleus. So we modified our profiling tool so that instead of looking at whole cells, we segment individual nuclei. So here for this workflow example, we're looking at SE35, either exposed to fat, growth, or bone conditions in 72 hours. So once we segment each individual cell, we acquire a set of descriptors. So traditionally, we mainly focus on shape-based and intensity-based descriptors, but now, collaboration with Dr. Mataguchi's lab, we're able to gather a new set of texture hairlet-based descriptors that can better describe the organization of these reporters within the nuclear space. So once we gathered all of these descriptors, for the third step, we sought to identify differences in these descriptors across the conditions. So here we just look at one descriptor, which is relative fluorescence, and we look at our 6 reporters. For laminin, HTTP4, RUNX2, and EZH2, we see no statistically significant differences. However, for nuclear mitotic apparatus and NUMA, we found that there was a drop in the expression of cells exposed to the osteogenic condition for 3 days, and SE35 domain was not increased in the osteogenic for 3 days. And then if we take a closer look, we see that NUMA, if we apply our modified profiling approach, and then reduce the many descriptors to 3 dimensions, we see that we can discriminate between the 3 conditions. And we have a high sensitivity, specificity, and accuracy of our classifier. And then similarly with SE35, we also had very good separation. And then if we just look at RUNX2, for example, we saw that the differences, we saw a low accuracy between the different conditions. So the next question is, what makes NUMA and SE35 domains so special from the other reporters? So we look at the potential biological function of NUMA. It is involved in mitotic spindle formation during mitosis. It is also necessary for asymmetric cell division. And asymmetric cell division is necessary for cells to become different phenotypes when they're proliferating. And there's also, it's very, very abundant in interphase nuclei, which, like I said earlier, suggests that there's a potential role in the NUMA as a potential, basically as a responsive to outside signaling events. Then SE35 resides in the intrachromatin space, and it's a indirect measure of global chromatin changes, and it also hosts the machinery necessary for post-transcriptional splicing. And more specifically, it resides in between chromatin. However, in the periphery of SE35 domains, there tends to be more euchromatin or open state chromatin than heterochromatin, and several studies have cited that there is an overlap between euchromatin regions that host genes for adipogenesis, osteogenesis, and myogenesis. So, in conclusion to the first aim, we developed a nuclear-based single-cell profiling tool to identify, and this is good because we can identify single-cell complex cultures. And I'd like to mention that this workflow, even though we just looked at 6 proteins, we could look at many, many more reporters beyond the study. That being said, though, we identified 2 nuclear reporters. NUMA, which is a cell cycle regulator, tends to be potentially useful for classifying cell types, and SE35, which hosts euchromatin or open chromatin neighborhoods, and could potentially serve as an identifier of future biomaterial-induced stem cell responses. Brings us to our second aim, which is to apply the toolbox that we developed in aim 1 to identify heterogeneity in different stem cell-related contexts. So here, this is an interesting study where we have a mouse embryonic stem cell colony stained for different markers. Here at the top left, the embryonic stem cell colony is stained for OCT4, which is a pluripotent marker, and we see no differences between the staining, which would suggest that this is a homogeneous population. However, if we co-stain it with NANOG, which is another pluripotent marker, we start to see differences in the fluorescence between one cell and the other. And we overlay these channels with DAPI, which stains DNA. We see this nice collage of different colors, which shows that what we thought is a homogeneous population is actually very heterogeneous. And this is particularly problematic for biomaterial screening tools, such as this high-throughput screening chip here, where every single square here is a particular test condition. And if the cells in this test condition look like this, however, we get one readout, that readout could be highly misrepresentative of what's actually going on in that condition. Which brings me to the objective of this aim, which is to identify subtypes and heterogeneous cell groups. So first we decided to identify differentiating cells versus pluripotent cells and embryonic stem cell colonies. So here we have an embryonic stem cell colony, and then if we snap an image in the periphery, and then we stain it for OCT4, which is a pluripotent marker, we immediately see that cells closer to the center of the colony are pluripotent, and cells towards the periphery of the colony are differentiating. So then we decided to, now that we have these 2 groups, we asked the question, can we use NUMA descriptors to identify morphological differences between these 2 groups? So for each of these groups, we also co-stain with NUMA, and then we stain the cells, and then we gather the descriptors, and then reduce them to a 3-dimensional space, where again, each principal component represents a linear combination of the descriptors. We're able to parse or identify differences with a high accuracy between cells that are high in OCT4, meaning pluripotent cells, and cells in low OCT4, meaning spontaneously differentiated cells. So we did a similar study, but in this case using induced pluripotent stem cells. And now we look at SOS2, which is another pluripotent marker. And again, we ask the same question: can we identify these differences in this more harder to identify scenario? Because in the previous slide, it was qualitatively you could tell the differences. So here again, NUMA was able to give us morphologic differences between two conditions. So these findings are very exciting because we're able to identify pluripotent cells between differentiated cells, meaning in a heterogeneous group, we can identify cells that we want and cells that we don't necessarily want. However, one limitation of this work is in order to get the descriptors, in order to do our profiling, we have to fix and stain the cells. Hence, we're killing the cells and we can't really use them, which motivated us to try to apply a similar approach to a live cell platform. So, to do that as a proof of concept study, we labeled mesenchymal stem cells or adult stem cells and cancer stem cells with a live actin reporter, and then we decided to co-culture it. So, qualitatively, our eyes can classify differences between these two groups, and our classifier is also capable of doing that. For here, the red dots represent mesenchymal stem cells, the blue dots represent cancer stem cells, and there's a very nice separation between the two groups. So then in co-culture, decided to ask, we can identify differences using our classifier, using the naked eye, but can we do something about this live culture? Can we purify or remove cells that we don't like? So here, I'm going to say the bad guys are the cancer cells. So we want to remove them. So by doing confocal laser ablation, we're able to remove the cells from culture, and then as long as 20 minutes or longer, we see that the cells that we want are still in the culture, active and alive. So that was our proof of concept study. So we wanted to see if we can do the same workflow, but in cell cultures that are indistinguishable. So here we looked at human embryonic kidney cells, and we either treated them with etoposide, which induces DNA damage, or had a control condition where we did not induce DNA damage. So here's a phase contrast image of the two. Qualitatively, you can't really tell the differences. And then we introduced an emissary NUMA reporter, so we labeled the live cells with their NUMA protein, and then for tracking purposes, we also labeled the control condition with GFP actin. And then this represents a snapshot of a co-culture. So we co-cultured these two cell subtypes in a 50-50 mix. And here the pluses represent cells that were treated with etoposide or are DNA damaged, and the minuses represent cells that are not. And then we gathered morphometric descriptors of the NUMA protein for these two different groups and identified morphologic differences using a profiler with a high accuracy. So then the next question that we decided to ask is, if we remove one of these cell subtypes, can we affect the future outcome of the population viability? So to do that, we found that paclitaxel or Taxol this is an FDA-approved cancer-fighting agent. We optimize the concentrations of Taxol such that if cells are treated with etoposide or DNA damage, the majority of the cells will die off. Here, as shown here, where the blue represents nuclei. However, if healthy cells are exposed to the same dose of Taxol, there's minimal or no change in population viability. So, by introducing Taxol, decided to employ the following workflow. So first, we use laser ablation to remove healthy cells, and then we counted the cells, and then we treated the population with Taxol. So one would expect that the majority of the population should die off because the majority of the population is DNA damage. And then we count the cells again. So the control condition, which has a 50-50 mix of DNA damage and no DNA damage, saw about a 50% drop in viability, which is expected because Taxol killed about 50% of the cells. However, for the treatment group where we killed the healthy cells, we saw a substantial drop beyond the control condition of further 25%, which suggests that the cells, or the majority of the cells, are unhealthy, died off compared to the control. So, in conclusions for this aim, we found that NUMA can classify subtypes and heterogeneous cell cultures And this may be due to the potential biological role this could be that we are identifying differences in protein descriptors in the interface nuclei. And then we apply this to two different contexts. So first we identified proliferating versus differentiating stem cells, which is could eventually maybe be used as a potential tool to characterize new stem cell harvests. So if we know, so when we receive stem cells from different donors, there's usually a rigorous characterization process, and maybe we could implement this as an additional characterization technique to assess the heterogeneity of the population. And then we also apply this in a live system. And this could also set the basis for novel cell culture purification platforms, where we can purify live cultures, which render stem cells as even more attractive for different therapies and research. So now we're going to switch gears and wrap up the talk with the third aim, which we'd like to utilize our toolbox to classify biomaterial responses. So the experimental design here was we cultured mesenchymal stem cells in material conditions of interest, and we looked at many different platforms, but for this talk we're going to focus on topography. And then at 72 hours, we stained the cells with different proteins of interest, and here I'm going to focus with S35. And then we repeat the same exact experiment, same culture conditions, but carry it on to 2 weeks, and then we fix and stain with phenotypic markers. So here, for example, is alkaline phosphatase immunostaining, where the blue represents bone cells, and here we have a stain for triglycerides or fat cells. So then the question or objective of this aim is, can we identify a subset of descriptors at 72 hours that is predictive of future phenotype that is caused by a biomaterial condition of interest? So the first set of topographies that we looked at were a set of surface topographies, which in this work was done in collaboration with Dr. Murphy at the New Jersey Center for Biomaterials. And here we use the principle of phase separation, where we take two immiscible polymers and we mix them, and then we make a film out of them. They will phase separate, and then if we introduce an agent that can remove one of the polymers away, then we will be left with one polymer and the underlying surface beneath. So by doing that, we developed, by varying the ratios between poly-DT carbonate and polystyrene, we're able to develop discontinuous surfaces. So here we have a surface of, we have a film of poly-DT carbonate, and then here we have little pits, where at the bottom of the pit we have glass, and then we have a much lower ratio of poly-DT carbonate to polystyrene. We're able to develop little bumps or islands, where here the islands are made of poly-DT carbonate in a glass underneath. We refer to these as discontinuous. And then by optimizing the concentrations, we're also able to fabricate highly reproducible continuous surfaces. So here we have a phase continuity of the polymer. And these are representative images of actin. So we saw that if we culture stem cells under these three conditions, either two-dimensional flat, continuous surface topography, or discontinuous, we saw that in the continuous surfaces, we saw statistically significant increase in alkaline phosphatase at 2 weeks compared to the flat control. And in contrast, for the discontinuous surfaces, we saw a significant decrease in alkaline phosphatase response at 2 weeks compared to the control. So the question that we saw back then is, can we use our 72-hour S35 descriptors to identify differences between these three conditions that give you long-term phenotypic divergent responses? So we took all of the descriptors and then we reduced them to three-dimensional space, and then we were able to get a good separation between cells cultured in the continuous condition, which is blue, discontinuous, which is black, and the flat control, which is red. And here we looked at each individual principal component and then used a decision tree classifier based on the principal component, and we were able to, for each individual condition, get very high classification efficiency. So this is a very cool study in my opinion, because we're able to show that for three distinct topographies that give you three distinct long-term differentiation outcomes, we're able to identify these differences in stem cells much earlier on than any phenotypic marker available. So, moving forward in the same, the next question that we sought to ask was, can we use the SC35 organization that is particular to conditions, but instead of looking at just 3 different conditions, look at a subset from a much larger library. So, for this work, we formed a collaboration with Dr. Jan and his lab at the Tissue Regeneration Group at the University of Twente in the Netherlands, where they developed a high-throughput screening platform where, by developing an algorithm of different combinations of primitive shapes, they were able to develop thing called a total chip, where within one chip, which is 2 centimeters by 2 centimeters, it's about the size of your thumb, there's over 2,000 unique topographical conditions that we can test within one experiment. So this is kind of a snapshot or a bird's eye view of the total chip. And then if we look at SEM micrographs, we see that they are separated. There's distinct boxes or compartments that are separated. So here these microfillers are about 10 microns in height and separated by walls that are 40 microns. And then if we look at a more microscale view, we can have many distinct topographies next to one another, and then we can assess stem cell morphologies and responses, many conditions within one experiment. So the objective of this project was to identify early descriptors that precede different alkaline phosphatase expressions induced by the topographies. So Dr. DeBoer's lab did many screens, and then they identified a small cohort or subset of topographies that give you an increased alkaline phosphatase expression without the presence of bone growth factors. And because the actual topographies are proprietary, it's going to refer to them as total A through total D. So here independently, we repeated their experiments with the topographies identified, and we also found that in the absence of bone growth factors, we can identify a high level of alkaline phosphate cells. And we also found a decreasing trend at 2 weeks. And however, at 72 hours, the SC35 morphology is qualitatively can't really tell the difference apart. So the question that we asked was, can we, with this small subset of alkaline phosphatase inducing topographies, can we identify morphological differences much earlier on? So here we look at topo A versus topo D. We identify morphological differences at 72 hours, and similarly at the more subtle difference conditions, topo B, topo C, we also identify differences. And then if we look at all of the topographies collectively and put them in 3-dimensional space, and then we get the centroid of the different cultures, here we can get a rough estimate of the differences between all of the conditions with respect to one another. So, in conclusions for this third aim, we found that SC35 organization precedes long-term topography-induced differentiation, and there's 2 potential mechanisms that could be the cause of this. So the first one, it could be due to the euchromatin neighborhoods and the co-localization of these regions early on for genes that are necessary for the long-term differentiation outcomes. Or it could also be due to mechanotransduction, since SC35 resides in the intrachromatin regions. And the reason that we looked at topography is because in mesenchymal stem cells, they tend to differentiate towards connected lineages, and one of the more attractive of those is bone. And it's been shown that stem cells preferentially become bone on stiffer materials. So within the realm of stiff materials, one of the attractive additional parameters to be studied is topography, which motivated us to study topography. And another key finding here is that we were able to identify a subset of topography from a much larger topographical library with very fine differences in alkaline phosphatase expression. Hence, this has the potential, or we have set the stage or a potential tool to automate and classify many, many conditions within one experiment. And so that's the end of the third aim. So going to quickly just summarize my talk. And here we show that we developed and improved single cell profiling tool to advance the utility of stem cells in research and therapy. And in the first aim, we developed a nuclear-based profiling tool and identified that pneumoprotein and S35 chromatin domains are viable nuclear reporters. In the second aim, we found that pneumoprotein was capable of classifying cell subtype between pluripotent versus differentiating cells and DNA damage versus healthy cells in live cell cultures. And lastly, in our third aim, we found that S35 intrachromatin domains can predict topography-induced stem cell differentiation, both in a small subset of unique topographies and also in a subset of a much larger topographical library. So, moving forward, there are several directions that this work can go in. So the first one is implement this in a label-less real-time imaging. So even though we found that we could do live cell studies, we transfected those cells. So we introduced sometimes even viral reporters to the cells. However, we started a collaboration with Dr. Sistrom's laboratory at NIST, and here they developed a platform where they can digitally stain cells in real time, live cells, without actually perturbing the cells at all. So the way this works is they found, so let's say this image here is 10 pixels by 10 pixels. Within each pixel, they gathered a spectral signature, and they compared it to a training set of spectral signatures and deduced whether we're looking at lipids, the cytosol, nucleus, or mineral. So if we can combine these type of methodologies with our more high content throughput for these digital stains, then we can potentially have the same outcomes, but in live cells without perturbing cells. Another potentially attractive direction is to apply nuclear profiling for a high throughput cell sorting system. So conventional flow cytometers are able to separate or bin cells in different bins based on fluorescence. And as we saw earlier, fluorescence could be highly misrepresentative of heterogeneity or homogeneity in culture. Hence, if we instead or in addition to looking at fluorescence, we image each individual cell and then sort them based on nuclear profiling or a larger subset of descriptors, we could bin one's heterogeneous culture into two homogeneous cell subtypes. So it'd be kind of like an improved next generation flow cytometer system. And the third future working direction is something that we have been working on for some time, particularly with Dr. Androulakis, which is to figure out or elucidate the mechanistic role of these nuclear reporters. So, as we mentioned earlier in the talk, the nuclear space is affected by actin. But if we take a closer look to actin dynamics, actin is closely affected by Rho-A. So if we have GTP or activated Rho-A, it will induce the formation of stress fibers, fiber contractions, and hence will result in changes to the descriptors that we have been looking at. So up to date, we have identified a function of descriptors that gives us cell state. So in order to get the mechanistic role behind this, if we incorporate the GTPase expression of GTPases such as Rho-A or its antagonist RAF1, then we can have a more complex function that is more descriptive of what is going on, both temporally and in terms of outcomes. So real quick, my talk, a lot of the talk was summarized in these three publications here. So this one was published in Journal of Molecular Screening, and this one here is also based on the pneumoprotein. We're preparing to submit to +1, and all of the S35 work that I presented today, we are preparing to submit to integrated biology. And these were additional publications that I didn't have a chance to discuss here, but they resulted from active collaborations and other projects. That being said, I'd like to acknowledge my committee, particularly Dr. Mogi. Thank you for giving me the opportunity to spend these 6 wonderful years in your lab. I learned so much, and it's really been amazing. And Dr. Cohen, my co-advisor, I thank you. You gave me the opportunity to present at some of your group meetings where I received a lot of positive feedback, and I think it's helped me become a better speaker. And Dr. Interlakis, I'd like to thank you for the efforts in the systems biology work and for also serving as my committee. Not to Serrano, you are the expert of nuclear proteins, and the few discussions that we've had have been very fruitful and have been insightful as well. And Nogue Lab, I'd like to thank everybody here, particularly Varun. You've been by my side since my first year here.
And since his middle school.
Yeah, first year of high school. So since his first summer in high school, he's been coming every summer to the lab. Well, I've known him practically my whole time here. And then he joined the biomedical engineering program at Rutgers, and now he is one of our star undergraduates, and he's been very, very helpful. And Parth also, and Nandika, you joined us recently, and it's been great working with you. It's really been nice, and yeah, I mean, everybody here has done a lot for me, and it's been great. And the Cohen Lab, I'd like to thank Dr. Jared Bushman. We did some work with identifying cell subtypes ex vivo using oligodendrocyte precursor cells. Unfortunately, I did not have the time to present that here, but it was very exciting work. And Dr. Sanjeeva Murthy for allowing me to work on several projects, particularly with the surface topographies. And Dr. Sven Sommerfeld for just helping me with my dissertation document, particularly the sections of biomaterials. And the Bohr Lab, I'd like to thank Jan for giving me the opportunity to go to the Netherlands to pursue this project using a hacker platform. And everybody else, and the BME staff, just everybody collectively for just keeping things running and just keeping the building in one piece. There's a lot going on, and they've really done a tremendous job of keeping it together. And definitely the funding sources, because without the funding sources, none of this could happen. And last but definitely not least, I'd like to thank my family and friends. And yeah, I mean, these 6 years, it's been a lot going on. And so here's my family, and my mom, dad, they're back there, and my brother, my sister-in-law, and my sister. And they came all the way from Pittsburgh to watch me, and I'm tremendously appreciative of that. Yeah, I love you guys. And yeah, and all the friendships that we made here, it's been really amazing. Here's just some pictures of some friends from the Mogi lab and other labs. Here's some buddies who work with Samsung, and here's my brother and sister-in-law getting married, and these are some very close high school buddies. And they've all helped in one way or another. And that being said,
okay, very good. Questions from the general public first, please. Sure, you have questions, don't you?
yes.
Daniel.
When you're doing your laser presentation, did you like find each of the cells individually and like adapt it, or just
Can you repeat the question?
So the question was, how did we exactly do the laser ablation where we had the 2 cell subtypes? So what we did was we zoomed in at the nucleus of the cells that we wanted to ablate, and then we exposed it to a multi-photon laser several seconds, and that resulted in the complete ablation of the cell. And since we zoomed into the nucleus, it did minimal perturbation to the surrounding cells.
How do you know that?
Well, in our proof of concept study, we found that we looked at it for up to an hour afterwards, and we saw that the surrounding cells were minimally affected. I mean, they did change their morphology a little bit, but we did not see any apoptotic events or anything of that nature. Any other questions?
Any more questions? No more? Okay, thank you all for coming.
Yeah, thanks for coming, everyone.